Yeah, I did take the one in people there.
Okay. What time is it?
31.
All right, so we roll it. Oh, some people are. Okay. Go.
Barrel.
All right, can everybody hear me okay? You can hear that? All right. Let's see, one more person. Let's get the show on the road. Hi, I'm Melissa Runge-Morris, and I'm the director of the CURES Center. It's our delightful pleasure to have as our special guest seminar speaker, Dr. Trevor Penning from the UPenn, that's University of Pennsylvania, the Department of Systems Pharmacology and Translational Therapeutics. Dr. Penning is really a thought leader in our field. He received his bachelor's degree and Ph.D. degree from Southampton University in the UK. And he tells me that when he came for a postdoctoral fellowship to Johns Hopkins University, he never left. He stayed in the United States ever since that time. Dr. Penning rapidly rose through the faculty ranks at UPenn and has come to wear many senior hats in that busy beehive of activity. He's the face of environmental exposure science and environmental justice. Dr. Penning is the Thelma, Brown, and Henry Charles Molinoff Professor of Pharmacology. He's the director of an environmental health sciences core center, P30, called the Center of Excellence in Environmental Toxicology, and he's been the director since its inception in 2006. He's the face of environmental exposure science. You know, Philadelphia is an urban center with environmental justice problems, just like the city of Detroit. And Dr. Penning's passion is trying to understand the role and complexity of air pollution as a determinant of health consequences. We're going to hear more about his interest in air pollution and the contribution of diesel exhaust to air quality, and the metabolic activation of nitroarenes in air pollution that lead to adverse health consequences, including lung cancer. The title of his talk today is Air Pollution, Lung Cancer, and NRF2. Welcome, Trevor.
Thank you, Melissa. Thank you, Melissa, for that very warm opening. You gave me a tough act to follow, I think. I will try and wear the proper face moving forward. So thank you for inviting me. It's been a lovely time here, even though it's been brief. So today I'd like to tell you about a story we've been working on for several years now, and it relates to the concept of air pollution and lung health, and how we can start thinking about how we can use exposomics to look at this problem, and also our work on how this might be related to the activation of the transcription factor NRF2. So it was the International Agency for Research on Cancer in this monograph 109 that actually identified outdoor air pollution as a group 1 human carcinogen, meaning there is sufficient information to indicate that exposure to air pollution will cause cancer in humans, and the dominant form of cancer being lung cancer. When you think about lung cancer, you often think about cigarette smoking, and it's clear that about 85% of lung cancer is caused by smoking combustible cigarettes. But what about the other 15%? A large portion of that is related to air pollution, and the World Health Organization estimates that over 250,000 deaths occur globally per year to lung cancer from air pollution. So if it's a group 1 carcinogen, what does that really mean? This is the International Agency's research on carcinogens definition. If an agent is carcinogenic in humans, when there is sufficient evidence for carcinogenicity in humans and when there is less than sufficient evidence, there has to be sufficient evidence in experimental animals. And most importantly, the mode of action must be operational in humans. So if you look at the air pollutants that we can be exposed to, they fall into 2 categories. They fall into the black ones shown here, which fall into the class of volatile organic compounds. And you can see compounds such as 1,3-butadiene listed here. And you'll see they're ranking individually as group 1 carcinogens. The ones in red are ones found in particulate matter. And you will see that in that group, you have benzoylpyrene, which is the only one that has a group 1 designation. And then you have other polycyclic aromatic hydrocarbons, as well as the nitroarenes, which we'll be talking a little bit about later. So like everybody, our lab was closed for almost 18 months. And because of our interest in air pollution and lung cancer, we felt, what can we do that would be meaningful when we had no wet bench capability? So we started thinking about how we could do exposomics using public health information and relate this to incidence of disease. So we use several different methods to do this work. One area of public health information is known as the Toxic Release Inventory, TRI. And TRI is actually emissions by point source. And by point source, you also get speciation of the chemicals that are released by that point source. And knowing that there is a lag phase between exposure and especially lung cancer of maybe 20 years, we felt that we should go back over 30 years of data to generate heat maps for different exposomic features that might actually impact incidence of lung cancer. So I can't read this that well from here, but you will see that we have different exposomic features listed here, and these are aggregate maps over 30 years for the area of the metropolitan area of Philadelphia. And in the lower quadrants, you will see NASA satellite imagery data for PM2.5, which is fine particulate matter, and we've already talked about the carcinogens present on that. And that's at a 1 kilometer grid scale. And to the right of that, a map for nitrogen oxide gases, which is a surrogate marker of follicular exhaust. So we took these different exposomic features and combined them using a raster method, using GIS, ArcGIS, and came up with a cumulative map to show where the areas of air pollution were the most present over that 30-year period. And you can see that there are a series of spots lighting up that are linked almost one to the other, and that's the I-95 corridor. But you might think that if you live near an I-95 corridor, you have an equal chance of getting these exposures. And you can see there are gaps. It's not a continuous line. There are certain zip codes which are indicated there that are more prevalent than others in terms of the amount of cumulative exposure that has been experienced. Yes. Yeah, absolutely.
All of your point sources there have the same thing.
So I'm not sure if I fully understand your question, but what I'm saying is that these are chemical exposures from the point source of which we have geolocation. And what we've mapped them to are the various zip codes, right? So
just on the figure, every point source you have has the same diameter. It's got the same area of where you would live that you're exposed to. It's not like there's no small point sources, there's no big areas. They're all the same.
maybe we can talk about this later because I don't fully understand your question and I don't want to take up time on this. We use a different approach to this. And this is an approach used by the EPA. It's called the Modified Multiple Criteria Decision Analysis. And in this particular model, what one does is take into account the toxicity of the substance. In parentheses, I have IARC ranking. Persistence, meaning whether it's a volatile compound or not, which is a binary scale of 0 or 1, depending on whether the compound is non-volatile. And occurrence, which means the presence of that compound over all other compounds in the emission. And when we looked at the speciation in the toxic release inventory, we found there were over 230 chemicals that were actually present depending on point source. And for many of those chemicals, there is no IARC ranking for those compounds. So how could we determine then perhaps toxicity? So we wrote an algorithm to search all of the PubChem and PubMed databases to actually search for those compounds in terms of whether or not they met any of the 10 criteria that are linked to chemical carcinogenesis. This was a technique originally introduced by Martin Smith at the Berkeley Superfund program, and he applied this to benzene. So we took that example and applied it to 230 chemicals. And you can see that the 10 criteria there include things such as, is the compound electrophilic? Is it genotoxic? Will it cause inflammation? And the more of these hits you get, the stronger the idea is that this compound could be carcinogenic. Now, we weighted our literature search in ranking these compounds if the literature cited had a human component to it, and whether or not there was data to show the compound was tumorigenic. So by coming up with this method, we ended up with a single number score for toxicity for the 230 compounds. And then we included in the calculation the persistence and the occurrence, which I've already described to you. And this gave us hazard indices as single numbers by zip code for this exposure paradigm. And when you do this, you'll see a map very similar to the one that we had before in terms of the zip codes that were encountered in terms of these exposures. And it's always nice when you use two models and they come up with very similar data. So if you read this, it says the hazard index for 421 zip codes was calculated and 218 zip codes had a hazard score that went all the way up to 21. And you'll also see that many of these high-scoring zip codes actually were along the I-95 corridor, and the zip codes with the highest score weren't necessarily those with the highest emissions because we're taking into account the carcinogenicity of these compounds. So the next thing we did in this study was actually to look at the incidence of lung cancer in the Philadelphia area. And the average incidence of lung cancer in Pennsylvania is 53 cases per 100,000 individuals. And that's what you see in yellow. As you go up, you'll see that the incidence rate increases significantly in terms of the blue color. And the blue color you'll see is about 3 times higher than the average incidence. And you can see that in these two maps side by side here. We then wanted to ask whether there was an association between lung cancer cases and our hazard indices that we had calculated using the method so described. And you can see that there is a correlation between hazard index and lung cancer incidence. And this is no small sample set because we looked at close at 70,000 cases over a 30-year period and also took into account the exposure data over the same time frame. So there is a community outreach component to this, and that is now we have these hazard indexes for these 421 zip codes. We can start communicating these findings to communities and actually perhaps encourage the uptake of lung cancer screening. So now come back to some bench science. And so we know that nitropolycyclic aromatic hydrocarbons are a component of the particulate matter of air pollution, and they're most strongly found in diesel exhaust emissions. And I'm showing you that table again to show you where the nitropolycyclic aromatic hydrocarbons sit vis-a-vis the grouping by IOC. And in the right-hand side, you will see that when you start doing Ames test experiments to look at the number of histidine revertants you will see in salmonella, it's the fraction that contain the nitro pH that give you the highest signal. So when we talk about nitroarenes, it's a class of compounds, it's not just one. And you can see them all listed in this table here on the left-hand side. And one of the compounds that attracted our attention early on was this compound 3-nitrobenzanthrone. And in an Ames test, side by side to benzoylpyrene, it's 20,000 times more mutagenic. And that comes to the idea of how is this compound metabolically activated. So it undergoes metabolic activation by nitroreduction. And the nitroreduction requires 3 2-electron reduction events to take place. You go from the nitroso compound to the nitroso, you go from the nitro compound to the nitroso compound to the hydroxyloamino compound, ultimately to the amine. So it's 3 2-electron reductions from the nitro group to the amine. And as you will see later, the hydroxylamino compound is the one we're most concerned about. So one of the reasons this caught our eye was we were concerned about what are the enzymes that do this nitro reduction. And so a paper had appeared looking at the metabolic activation of this cancer chemotherapeutic agent, PR140A, I think it is. I can't read it from here. But anyway, what you will see is that this compound goes from the nitro group to the hydroxylamino group. And when it does that, that is the warhead on the molecule that does the harm, because that actually is a group for conjugation and release of a leaving group that can cause DNA adduct formation. And it was felt at the time that the enzyme responsible for this might be NQO1, an ADPH P450 oxidoreductase. And to look at this, a number of different candidate enzymes shown here were transfected into these HKT1116 cells. And this shows the transfection of the different enzymes, and they were all tagged. And this shows the ability of these enzymes to convert the nitro compound to the hydroxylamino compound. And here's NQO1 here. But look at what happens with this aldo-keto reductase 1C3. It's much, much higher in terms of its ability to do the nitro reduction, which was unexpected at the time. And in this corner here, we actually have a measurement of the enzyme activity in the transfected cells using an artificial substrate, puberone. And here is AKR1C3 here. So let me tell you about the aldo-keto reductases then, which is now implicated in the nitroreduction of these nitroarenes. So this is a gene family that we have been working on for more than 30 years. And these are mainly monomeric NADPH dependent oxidoreductases. And what they like to do is convert carbonyl groups to alcohols. So an aldehyde will go to a primary alcohol, a ketone will go to a secondary alcohol. These are monomeric enzymes in the whole. They're cytosolic, and they're present in all phyla. And there are 173 protein members that fall into 16 families. There are probably many, many more that are only characterized in terms of their presence in genomes, but they have not been functionalized in terms of expression. Now, their substrate specificity is broad, and you can look down this list here and realize there are many compounds that have aldehydes or ketones within their structures for which these enzymes can work. Sugar and lipid aldehydes, steroid hormones, prostaglandins, and in some chemical carcinogens as well. But none in those chemical carcinogens are the nitroarenes. To the right, you will see a structure of aldo-keto reductase. It was the first structure of a human aldo-keto reductase done. It was done in my laboratory. I won't let you look at the date too much because it was done 30 years ago, but it's done by a graduate student. And this structure is known as the Tim barrel, where there is an alternating arrangement of alpha helix and beta strand that repeats itself 8 times. And the beta strands make up a barrel, which you can see in the middle. And at the bottom of the barrel is the catalytic tetrad. Now, the intriguing thing about these enzymes is they like to do 2 electron reductions. They like to take a hydride and put it on a carbonyl. Yet, if they're going to do nitroreduction, they have to do the reduction 3 times. So that was really unexpected. So the aldo-keto reductases, you've seen some of them already, and the ones in blue are the ones that we're most concerned about when it comes to perhaps nitroreduction. In humans, there are 15 members of the aldo-keto reductase family. We have them all in the lab with recombinant form, but we'll be focusing on the 1C enzymes, and you will see that their natural substrates happen to be steroid hormones. So we have the enzymes, and these are the nitroarenes of interest that we would like to study. And we're going to be focusing, as I said before, on 3-nitrobenzanthrin. So this is the proposed mechanism then of the metabolic activation of nitrobenzanthrin. You start off with the nitro compound here. You go to the nitroso, the hydroxylamino, and to the amine. And there's those 3 2-electron reduction steps you have to do. And I remind you that the compound of interest is the hydroxylamino compound, because this is a substrate for sulfur transferases and N-acetyl transferases. And once that gets put on that hydroxyl amino group, you have a good leaving group. And now you are set up for making DNA adducts, going through either the nitrenium or carbenium ion to form N2 adducts with deoxyguanosine or C8 adducts with deoxyguanosine. These compounds also have the potential in air to be oxidized back. And so therefore you have a way to generate reactive oxygen species from these compounds as well. So this is some of our first experiments that we did here. And this was a look using reverse HPLC to look at the conversion of free nitrobenzanthrone to the free amino compound. And you can see a nice peak of the free amino benzanthrone being formed only in the system that has the enzyme. And when there's no enzyme present, you see no transformation. In the bottom, you see a product-precursor relationship that's occurring between substrate depletion and product formation. We then looked at a panel, the complete panel of AKRs to see whether or not they could do this reaction. And you will see that the ones that are most prominent are the AKR1Cs, as predicted by the work done on that cancer chemotherapeutic agent. We were intrigued by the fact that we could do this 6-electron reduction. And so we wanted to determine whether or not we could see any of the intermediates. So using a high-resolution mass spectrometry coupled with liquid chromatography, we were able to detect the intermediates, the nitroso compound and the hydroxyl amino compound. And you will see at the bottom here, the MS and MS2 spectra to exact mass by high-resolution mass spectrometry of both the nitroso compound with loss of the carbonyl, and on the right-hand side, the hydroxyl amino compound with the loss of the hydroxyl group. We then compared these enzymes using steady-state kinetic parameters to see how they compared one with the other, and compared with NQO1, which had been the enzyme touted to be responsible. And as you can see, when it comes to catalytic efficiency in the table below, you will see that AKR1Cs have catalytic efficiencies very similar to quinone reductase. However, the major difference is in Km, in that Km for NQO1 is about 8, whereas the AKR1Cs are much lower. And that's exactly where you want to be, have a low Km enzyme when you're doing with a environmental exposure. And also, AKR1C3, 1C1, and 1C2 are all present in the lung. So what do we know about these aldo-keto reductases and how they're regulated? So we and others have shown that these enzymes are induced by the NRF2 system. So for those of you who don't know about the NRF2 system, let me just give you a reminder here. And that is NRF2 is a transcription factor that is sequestered in the cytosol by a KEAP1 protein, which actually brings it into juxtaposition to the coal-free ubiquitin ligase, which allows NRF2 to be proteasomally degraded. Therefore, the amount of NRF2 present in the basal situation is low. However, KEAP1 is a sensor for electrophiles, reactive oxygen, anything that reacts with a cysteine. And when that takes place, you disrupt the interaction of NRF2 with the colin-free ubiquitin ligase, and therefore newly NRF2 is no longer captured for degradation and makes its way to the nucleus, where it can bind to the antioxidant response element with its partner small math. And you will see that some of the proteins or genes that are expressed here is NQO1 and the AKR1C1 gene together. So I think about NRF2 in a dose response model, which shows a hormesis dose response, in which that too much and too little can have an adverse effect. So it's clear that if we don't have enough NRF2 around, we can't necessarily protect our cells from electrophiles, and therefore you will have a deficiency leading to toxicity. You will have a range in which it can be governing homeostasis. But too much NRF2 can be deleterious. Think about how NRF2 regulated genes are involved in cancer chemotherapeutic drug resistance, for example. So I believe this is a U-shaped dose response curve. So that gets into a conversation about the dark side of NRF2. So if we look at NRF2 and chemoprevention of initiation of cancer, we know that NRF2 induces detoxification enzymes. We know that the knockout mice are more susceptible to chemical carcinogenesis. We know that NRF2 activation prevents carcinogenesis in mouse models using NRF2 inducers. People have said that NRF2 is a tumor suppressor gene. And this has led to the concept that NRF2 inducers could be used in clinical trials to reduce the incidence of cancer. This work is ongoing in China right now. But we also know that NRF2 induces enzymes that result in cancer chemotherapeutic drug resistance. We also know that NRF2 is constitutively active in many cancers, and this is due to hypermethylation of the KEAP1 promoter and also mutation in NRF2 itself. NRF2, by trying to counter reactive oxygen, increases redox state and NADPH, which is an anabolic signal and can lead to tumor promotion. Others believe, therefore, that NRF2 is oncogenic. And that also suggests that there's interest in NRF2 inhibitors for cancer therapy. So what's the tipping point? When does one have too much of a good thing? This is the antioxidant response element in terms of its positional matrix analysis. And if one looks for the presence of this ARE in the AKRs, you will see that AKR 1C1, 1C2, and 1C3 have a battery of antioxidant response elements in their promoters. And although NQO1 is used as a surrogate of NRF2 activation, this expression with an NRF2 inducer is a measly 2 to 3-fold. With AKRs, it can be several hundred-fold, just to give you a barometer. That leads us to the following paradigm, and that is air pollution, by its very nature, will stimulate NRF2 and turn on a battery of AR-responsive genes. We also know that air pollution has nitro-pH in it, and those nitro-pH will be activated by the same genes. And during the activation process, we are going to produce electrophilic intermediates, and we're going to produce reactive oxygen species, leading to a feed-forward mechanism by which we could exacerbate our exposure to these compounds. So can we test this in some way? And so we have been actually thinking about working in 2 different cell lines. One is the wild type A549 human adenocarcinoma cell lines, which actually have constitutively active NRF2. They're a cancer cell line. NRF2 is presently in a very high situation. But this gives us the possibility, as you will see, to do CRISPR gene knockout of NRF2 in these very cell lines that have both a heterozygous and homozygous cell line. And then we can use human bronchoepithelial cells that have been immortalized with the KT approach, which means cyclin-dependent kinase has been engineered into the cells, as is reverse transcriptase for human telomerase to prevent cell senescence. So these cells are normal normal human lung cells, but have been immortalized by genetic engineering. And these cells are therefore can be responsive to NRF2 inducers such as sulforaphane and CDO, imidazole, whose structures are shown below. This shows formation of free amino benzanthrone in the two cell lines. In the left-hand side, you will see a reduction of free MBA to ABA in A549 cells. And you can see with constitutive NRF2 expression, that's a lot more robust than what you see in the HBEC-KT cell lines, which only have basal expression of NRF2. What's interesting in the lines for the HBEC-KT cells is the fact that there seems to be a curve that's lifting off, suggesting there may be some induction of NRF2 occurring. So this is our validation of our knockout of NRF2 in A549 cells. And you will see that when you go the heterozygous and homozygous cell lines, you see a lot less of AKL1C1, 1C2, and 1C3. In the homozygous cell line, they're completely eliminated. And at the bottom, you see the corresponding fact that even with a complete knockout, some NQR1 remains. And then on the right-hand side, you see a side-by-side comparison of those RNA measurements. You can also see at the protein level, so these are a series of immunoblots whereby we're looking at the expression of NRF2 in the wild-type cells, the heterozygous and homozygous, and you can see that NRF2 is completely lost in the homozygous, as is the expression of the AKL1C2 and 1C3 enzymes, and some NQR1 remains. So there's good concordance between the protein and the RNA measurements. So the way we measure free ABA formation in cells is actually to use a plate reader assay, in which we take advantage of the unique fluorescent properties of ABA, which are not present in the nitro compound. And then using this assay, if you look at the right-hand side, you can see how one can really have a significant effect on ABA formation in the heterozygous and homozygous knockout cell lines. So this tells us that ABA formation from nitrobenzanthrone is very much dependent upon NRF2. And by consequence, therefore, dependent on the AKR1Cs. So we want to do the reverse, in which we look at NRF2 activation in HBEC-KT cells. And you can see that with increasing doses of sulforaphane or CDOM, which are NRF2 activators, you can see increased expression of the AKR1C enzymes and NQR1. And then if you look at the comparison on the right-hand side, you can see how the AKL1C expression can very much dominate over what we're seeing with NQR1. And this is at the RNA level. We did similar experiments at the protein level, and you can see that when we have the inducers present, we see more NRF2 go to the nucleus than in the absence of inducer. And you can also see the increased expression at the protein level of the AKL1C enzymes. And that is quantified in the histogram to the right-hand side. And then looking at induction, in this case of NQR1, you can see the corresponding increase in enzyme activity with the two inducers. And we can then use that plate reader assay then to ask, do the NRF2 inducers increase ABA formation? And the answer for both inducers is yes. So I've told you that We have an idea how NRF2 is involved in the metabolic activation of these nitroarenes, and also indicated to you that there's a possibility that the nitro compounds may induce their own metabolism. So we were struck by the observation that compounds like nitrosobenzene, shown here, can actually form nitrosophiles, which can rearrange to sulfonamides and sulfonamides. And that made us think, well, maybe the nitroarenes, such as nitrobenzanthone, could directly modify the cysteine residues on KEAP1. So we actually did an experiment with recombinant KEAP1 and nitrosobenzanthone, the first 2-electron reduction product of nitrobenzanthone, and then used top-down proteomics to see whether or not we could see modification of those sulfhydryl groups. And the answer was yes. We can see covalent modification of KEAP1 with nitrosobenzanthone. I think this is the first example I'm aware of in the literature that someone has shown covalent modification of KEAP1 by a chemical carcinogen. To show you where those cysteines are, you can see those cysteines are actually have been entertained as being involved in the sensing mechanism of KEAP1 before. So I want to show you some unpublished data now. This data was generated by Anthony Zhu, who's a postdoc in my lab, currently on the job market. And he developed for us, for the first time, a mammalian mutagenesis assay, the hypoxanthine phosphoribosyltransferase transferase mutagenicity assay, in which this is a forward mutagenesis assay, in which you're looking at compounds that survive the treatment with 6-thioguanine. And on the left-hand side, you see the negative control, and on the right-hand side, the positive control that you see when you give a known mutagen in this assay, methane sulfonate. So he wanted to ask the questions, can the AKR1Cs increase mutagenicity in the HPRT gene mutagenicity assay? And what you will see is that when you have AKR1C present in increasing amounts in terms of the transfection and increasing amounts of 1-nitropyrene in this case, 1,8-dinitropyrene, and 3-nitrobenzanthone, all 3 nitroarenes, you will see a significant increase in the mutagenicity. This is the first time that we've actually seen direct increased mutagenicity driven by AKR1C genes in a mammalian cell system. So my summary is as follows. Geocoded hazard indices based on exposomic features derived for 420 zip codes in the Philadelphia metropolitan area have been generated. The hazard indices show a correlation with lung cancer incidence and can therefore perhaps increase the uptake of lung cancer screening. Compounds in air pollution, such as diesel exhaust constituents, are all activated by the AKR1C enzymes, which are induced by NRF2. Nitroarene activation is NRF2 driven, as shown by the CRISPR-Cas9 knockdown of NRF2. We know now that the nitroso intermediates can modify KEAP1. And we also know that KEAP1 will, through NRF2, induce AKR1Cs, and the AKR1C enzymes will increase the mutagenicity of free MBA. So we believe that NRF2 has a dark side in carcinogen activation in human bronchoepithelial cells. So some acknowledgements for the work. From my own laboratory, a few people have to be called out. So Jessica Murray was a Ph.D. student who now works for the EPA in the Inhalation Toxicology branch. She was responsible for the free nitrobenzanthrone work. Katia Marquez was responsible for the proteomics on KEAP1. It's a new postdoc in the lab. Anthony Zhu, you saw his HPRT gene mutagenicity assay. Tom McKeon for helping with the exposomic mapping of the Philadelphia metropolitan area. This could not be done without a group of really good collaborators. Clementina Mazaris in our mass spec core with guidance from Ian Blair. Wei Ting Wang for doing the correlation studies on exposomic features. Vogel Art for providing free nitrobenzanthrone metabolites. Karen El Bayoumi for giving us nitropyrene metabolites, which I haven't spoken too much about, and David Ross for recombinant NQO1. And the funding. So I thank you for your attention.
I was just wondering if you think that the prevalent modification of KEAP1 as a system is a physiological mechanism for turning off KEAP1, or is it like a successful artifact?
Well, we're doing those experiments in cells right now. I can tell you we have been able to see the KEAP1 modification in the HPEC-KT cells. It's an N of 1.
Is the modification that you see in KEAP1 on the system, does it limit to just the system or the tyrosine?
So it was bottom-up proteomics targeted for detecting those peptides with that modification based on mass difference. So I can't answer the question. Yes.
So Trevor, fantastic presentation.
Thank
you
you.
so much for that. Public health data is so valuable.
Yeah.
And I noticed you described the data as being age adjusted.
Yes.
I wondered if you could explain, was there not enough data to stratify it, or how do you age adjust such data?
Oh, sorry. So the age adjustment is the age adjustment for cancer, lung cancer incidence. That's the number that's available in the Pennsylvania Cancer Registry. They age adjust their numbers based on longevity of Pennsylvanians. Yeah, yeah, yes. Yes. Great question. So it was predominantly non-small cell lung carcinoma, but yes, yeah. Right. That's exactly what we saw. And which actually allows me now to show you the slide I've been waiting to show you. I planted you there. Right. So we asked the question whether or not air pollution might affect lung cancer survival. Right. So this is, again, a Pennsylvania lung cancer registry data. And you can see that it's over the same period of time, 1990 to 2017, 250,000 lung cancer patients here. And here we used EPA air quality system monitoring over a 24-hour average for each pollutant. And you can see the pollutants there are the priority air pollutants. And you can see whether this is related to whether the cancer is local, distance, or regional in its presence. And I want you to look at the shape of these curves. They're all U-shaped. You know, when you see a U-shaped dose response curve on an area of 2 and you see this in public data, it makes you start thinking that there is something really significant going on here relating to the mechanism that I've described to you. So thank you for asking the question. Yeah.
Yeah, I have a KEAP1 question and then
Okay.
So for the KEAP1,
Yeah.
Say it does increase the ubiquitination of KEAP1.
Yeah. Right.
How do you then tease out which of those mechanisms you want to work? Whether it's just a direct oxidative stress from the pollution towards this.
Well, I think the way we can get at that is through proteomics, because if the cysteines are being modified by reactive oxygen species, we can look for sulfinic and sulfonic acid derivatives of the cysteines. Here, we're actually seeing covalent modification by the carcinogen, which is really cool. Not if you are exposed, but it's cool from a scientific perspective. Yeah, AKR question? Yes. Oh, yes. Oh, yes. The AKR family of 1C1, 1C2, 1C3, and 1B10 light up in multiple cancers. And I think this is actually part of their response to chemotherapeutic drug resistance, which is a whole nother story that these enzymes are actually involved with. And early on, the AKR1Cs and 1B10 were considered part of a smoking gene battery, in that if you took buccal cells from smokers and then analyzed them while they were smoking and when they quit, you would actually find all these AKRs go up and then come back down. Yeah.
So for any of your cancer registries, you have genetic information or do something about you?
Not for the cancer registry, but it's a great question for the UK Biobank and a great question for all of us Biobank. Yeah. Yeah.
What about DNA?
Okay.
Talk about the adducts.
Okay. So there's two types of DNA adducts, covalent and oxidatively damaged DNA adducts. And we have a really sensitive stable isotope dilution LC-MS assay for measuring A-oxo-guanine. And we've already, I didn't put it in the talk because, you know, you have to select. I could have shown you a slide whereby we actually see dose-dependent increase in A-oxo-DG with 3-nitrobenzanthrone in A549 cells. We've done the experiment, it just wasn't in the talk. The covalent adducts are more challenging because although we can detect them, we do not yet have a stable isotope internal standard that we can use. And so we can detect them, but we can't quantify them until we have that internal standard, but we do see them. And when we do see them, and in a quantified way, we will then be able to, for the first time, be able to compare the relative abundance of those that come from reactive oxygen versus those that come from covalent modification. I have a sneaking suspicion the reactive oxygen species adducts are going to be higher.
Our community outreach co-leader, Terry Leach,
Yes.
question for you. She wants to know, have your findings been reported to the community? And if no, any plans? And if yes, can you say a word about the process and communication strategy?
It's a great question. So we thought long and hard about this. Before we disseminated any information to the communities, we thought about the ethical component. The ethical component is, on the one hand, you think community people have the right to know. They live in these communities, they are exposed to these hazards, and they need to know what their risk is. Downside of not thinking about this more carefully is that what would that do to their property values? What would it do to their access to health insurance? So we've been working with the Annenberg School of Communication to come up with a very thoughtful way by which we can communicate this data to the communities. And we've come up with a series of videos, which we are now pilot testing in an implementation strategy to see whether or not it really will increase people's desire to actually undergo lung cancer screening. There is a caveat. You know, people talk about lung cancer screening. This is the other thing that we're actually working on in the center, is that lung cancer screening has a 96% false positive rate. I don't know if you know that, but that's the actual fact. And therefore, to encourage people to get lung cancer screened and then see undefined nodules and then take them through an invasive procedure to determine whether those nodules are carcinogenic or not, has its problems associated with it, as you can see. So there's another effort going on in our center right now to come up with much better intermediate biomarkers of lung cancer. so that we can have a real stratification process to be able to get people into lung cancer treatment or screening if the risk is considered to be high. And so we need the exposomics data, which we have. We need new biomarker data, which we're working on. We actually have some really cool lipidomics and metabolomics data that can discriminate between different types of lung cancer. It's that information together that will ultimately be able to actually really have directed strategies to encourage people to take up screening and so on.
I'll follow it up with a related question.
Yeah.
Which I asked one, which is, what do you then feel is your responsibility for the government?
We've already given this information to the Philadelphia Department of Health. We've already also given it to the Air Management Services of Philadelphia, which is how EPA actually decided they weren't going to take responsibility for looking at the air quality in Philadelphia, but gave it to a private contractor, Air Management Services.